I'm delighted to be moderating our last panel of the day, and this panel is focused on what is normal and what is pathological. So we are lucky to have three leading lights in clinical grade variant interpretation, genetic variant classification, to help us understand how we currently separate pathogenic from normal variation in our genomes and where clinical genomics is headed. Our panel is focused on genetic variant interpretation, but I think many of the same thorny questions that we'll be discussing today apply to many other areas, or virtually all areas of medicine. And so this is far from an abstract academic exercise determining what exactly is normal. Variation may determine a genetic diagnosis, who's at risk in the family, and who's eligible to receive potentially life-changing therapy. I'm just going to quickly introduce our panelists, and then they will take it over for opening remarks. So first, Dr. Birgit Funk is a Vice President of Genomic Health at Sema4. Dr. Sharon Plon is a Professor of Molecular and Human Genetics at the Baylor College of Medicine. And Dr. Vandana Shashi is a Professor of Pediatrics at the Duke University School of Medicine. All three of our panelists have all led major research or clinical initiatives, and these have spanned the spectrum from understanding the genetic basis of cancer and inherited heart disease to interpreting genome sequencing for children and adults with undiagnosed diseases. I've asked our panelists to deliver opening remarks to help us set the stage for understanding the evolution of variant classification standards, the current state of the field, and where we might be headed over the next few years. So I'm going to turn the podium over to Dr. Funk now, and you can take this if you want to advance. Yeah. Yeah, so you can take this mic, and then the green should advance your slides. Yeah,
okay,
sure.
Thank you. It's a pleasure to be here today to debate this topic that always gets the blood boiling, at least in my circles. There are no two people in my field who cannot spend hours debating a variant. So without further ado, let me try to advance. Yeah, so what is normal, what is pathological? I mean, that really is a good question, and I'm not sure how much closer we are to the answer. But here's a bit of my perspective. So I'm a board-certified clinical molecular geneticist. I work in a clinical testing lab where we generate results, interpret them, and send them to the ordering provider. And from my perspective, things began to be complex when we began to sequence entire genes. Right? If you look at this evolution of genetic testing on the far left in the 90s, 80s, we were testing only what's known. So it was a little simpler. We would return your positive, negative, and that's it. But then in the 90s, we jumped over to begin sequencing genes, and now we started having this issue that we detected something that we didn't already know how to interpret. And with that, the discipline of variant science, as we call it today, was born. So this is my main slide. I'm going to bring the laboratory perspective to this panel. And one of the main issues that questions that's being debated is whose responsibility is it in the first place to do all this? It's more complex than you might think from a regulatory perspective. So you have these two entities here, the laboratory and the provider, and there's interaction. Like we generate the results, we send them over, the provider does something with them. In the old days, this is what we did. The lab ran the test, there was a result. We actually didn't do much interpretation because we already knew what the variant meant, and we issued the positive or negative report, and the provider then used that to integrate it in medical management. Today, it's very different. We have the same thing up front. We run the test, and then because of this novel variant detection problem, laboratories had to step up to the plate and now begin implementing steps to to interpret them. And I want to call out here, in case it comes up later, this is a two-step process, really. One is information gathering, and that's not to be overlooked. It's not simple. And this is the basis for what we call variant science. And then there is the actual classification, so applying frameworks that our professional societies are giving us. And with that comes now the unfortunate species of a variant of uncertain significance that has entered our practice and is causing so much headache. Now the provider takes this information, and there is not infrequently additional interpretation because the provider has the patient in front of him or her, additional clinical information that we may or may not have had in the laboratory, and there is often back and forth. Now, standardizing all this is extremely important, as you can imagine, and the main questions we have here are, well, the current standards are non-binding, really. I mean, they're out there, but if you implement them exactly as they're published, not much will happen. The question also becomes, should this be regulated actually under CLIA or not? And this is more of a debate, as you can imagine. There are active regulatory discussions going on all the way at the top. CLIA is being analyzed and up for modernization, and the body that's facilitating this and advising is organized by the CDC's CLIAC, the CLIA Advisory Council. So I've gotten invited to join these discussions, very interesting, of how this question is debated. How should CLIA be revised to clarify the role of the laboratory with respect to result interpretation? So very timely panel today. FDA is also getting interested in this and is beginning, from what I'm hearing, to mandate that clinical trials that are based on genotype base their genotypes on ClinVar, 3-star classifications, which are expert-driven. So that's my last one here. In addition to these 3 questions that were I introduced on the last slide, here's what keeps laboratory people like me awake. Terminology, as we introduced in the beginning. What does pathogenic even mean? And should it be used for low penetrance variants, which are also there? You just came out of this whole session of polygenic risk scores. Can a single word even accommodate the whole range of variation? Automation, always a fun topic. AI versus human experience. Where do we draw the line? Education, extremely important. Genetics is beginning baby steps to move out of the academic ivory tower. And with that, we really need to take care of our general practitioners and make sure that they have genetics expertise. And finally, the clinical report. So this is the product the laboratory issues and sends to the provider. What amount of detail needs to be on a lab report? Do we put the whole evidence there? Do people even care? How do we talk about it? Or do we just send them the bottom line? So these are just some of the questions we might debate together from my perspective. Thank you.
Well, that was a perfect segue. So hi, I'm Sharon Plon. I'm a medical geneticist, and so I'm going to take the physician's view. And I'm based in a children's cancer center, but I'm also in the Texas Medical Center, so I get lots of questions from all kinds of physicians. So I want to talk about Birgit's last point, which is genetic test reports are not only for geneticists. So if you really think about where we stand right now, there's at least 4, really 5 different audiences for genetic test reports. People ignore the lawyers, but the lawyers actually drive a lot of it. There's a ton of fine print so that the lab can't be sued, saying exactly what they don't pick up, what they could pick up, but they don't pick up, they're not guaranteeing. So the reports are full of that. There are the reports for the other variant experts. What is Heidi Rehm going to think of my report? You know, what is my colleague in another lab, if they get to see the report, going to think? And that provides, what if they see another member of the family? They need to know what evidence I looked at so they can look at what evidence they have, and maybe together we can solve the case. Includes things like algorithms, Polyphen, no offense to Shamil, who I think left, but his damaging description in Polyphen. I get lots of questions from physicians. What does damaging mean for my patient? And then there are medical geneticists and genetic counselors who I think most labs think are their primary audience. But to be honest, and I'm involved in ClinGen, so I know a lot now about variant classification, most clinical geneticists actually don't learn that much. And I think it's a little better for genetic counselors, but in my experience, it's not a ton better about how ACMG variant classification actually works. And then there's everybody else. There's every other physician and the family. And in my experience, non-geneticist physicians and patients actually have about the same amount of knowledge of medical genetics variant interpretation. And so you actually can write relatively similar reports for those two groups. So unfortunately, everyone else, this last group, are actually the people who are going to follow the patient over time. So even if a medical geneticist like me gets the report at first, I'm not that likely to see the patient 5 years later, but their general practitioner or their oncologist, if they develop cancer, are. And so it's important to remember that longitudinally, those are by far the most common people to see a report. So why do I bring this up? I think we are in a complete ongoing state of confusion, particularly because of the use of term variant of uncertain significance, but even if, and so these are real questions I receive on an almost daily basis from physicians, nurse practitioners who see reports that have a VUS on them. And often these are reports with no data to suggest pathogenicity. So I'll get questions, I'm in a pediatric hospital, child has a VUS and BRCA2, not even sure why it's on the report, but I get a question, should the mom have an oophorectomy? Just recently got a report, child with cancer with a VUS, single VUS and a recessive gene. Can the brother still be used as a bone marrow transplant carrier donor? These are important questions, right? Because maybe that brother has the same VUS too. I see the genetic test report was updated and now the VUS is gone, right? So a lot of labs, if a VUS is classified as likely benign, will now take the VUS off the report. That is very confusing, actually, to physicians. Like, what does it mean that it's now gone? And these are things that I think we as a field just really need to wrestle with, is how do we write reports that can last and can be used by the general medical community and by the families. And so I think we really do need new standards in reporting. So I'm on the committee, the ACMG, ClinGen, actually, as of today, I heard from Heidi, it's also CAP and AMP, are working together to update the variant classification guidelines. And we hope to make the basis of classification clearer and more quantified. But I actually think we need equivalent work on optimizing how genetic test reports are fashioned so that their meaning is clear across the spectrum of users to avoid the current state of confusion. And I am sort of a believer that we really need a top of the report, which very clearly says what we do or don't know, and could potentially just leave VUSs out of that part of the report entirely, or maybe, as Heidi mentioned, that we're going to subdivide them. And then the details for the geneticists, the lawyers, and everyone else could go on a second page or further down, because I really think this is an area that needs substantial improvement. I know there was a lot of back and forth about the All of Us program in the last session, but one of the things they've really done is done a huge amount of research on how do people perceive test reports, and it was sort of fascinating. They gave a talk, I think, at ACMG last year, and it was just fascinating, things that we think are completely understandable that were completely misinterpreted when you actually do research. So we need the kind of research that Amazon does on their webpage and things like that. Like, how do people actually perceive the reports we create to make them more useful? Thanks.
Thank you. So Dr. Plon referred to the mixed audience for genetic test reports, and among probably the biggest audience are geneticists and genetic counselors and people who practice genomic medicine outside of that specialty, neurologists, cardiologists, et cetera. But I would say even for people who are used to genetic reports, there are lots of questions that the community needs to think about that we still struggle with, be it in the research realm or in the clinical realm. I straddle both, and so this is where I think we have to have some more debate. So for example, we all know that ACMG and AMP have these guidelines for variant classification. Everybody's alluded to that, and many in this room are familiar with that. Labs will classify variants into 5 categories and will report 3 of those: pathogenic, likely pathogenic, and variants of uncertain significance. There is no analogous clinical classification of those variants. What happens when the clinician gets that report in their hands? How do they decide how they're going to communicate this to families and patients? So for example, do all pathogenic variants indicate a diagnosis? Not necessarily. There could be a variant that is a frame shift in a gene that you know is associated with loss of functions and can cause disease associated with loss of function, but you as a clinician might have information in hand that makes you decide that's not a diagnosis for your patient, and there are many examples of that in clinical practice. By the same token, do VUSs always indicate that the patient doesn't have a diagnosis? Not necessarily. You as a clinician may elevate a VUS into a likely pathogenic or a pathogenic based on your own assessment of the variant and the patient, because you have the patient at hand. ACMG guidelines leave room for the patient, for the clinician to interpret variants on their own, although the lab issues a report. In our own study, we found that in about 150 patients that we studied retrospectively, about 10% of the time, the ordering clinician disagreed with the laboratory classification of the variant. They did not either agree with the diagnosis or they upgraded a variant that was at a lower level into a diagnostic variant. And so this could happen across all the 3 categories of variants that labs report. And it's been known in the literature that about 3/4 of genetic clinicians, be it counselors or geneticists, they will reinterpret VUSs themselves before they give it to patients. VUSs can be subcategorized kind of informally into hot or cool VUSs, and clinicians these days are really good. Sharon is right. There's a wide variety in terms of the knowledge that geneticists have, but many younger geneticists and many geneticists who kind of go to all the conferences and update themselves are able to do kind of a classification themselves. So they may take this matter into their own hands. But that process is really not standardized. How do clinicians interpret variants? Some of these parameters that I've outlined in this figure are the factors that a clinician can take into account. So for example, what is the mode of inheritance of the disorder that your patient has? And does that match with the variant that you've seen on the report? Does it fit the phenotype of your patient? And this is really, really important. If it doesn't fit the phenotype, many clinicians will disagree with the diagnosis. And do you need additional evidence for you to say that this is a disease-causing variant, even if the lab has reported as a likely pathopath? So for example, do you need a biochemical test to confirm it? Do you need to do variant segregation in other members of the family for you to feel more confident? And lastly, the holy grail in all of genomic medicine is, can you really use these results for prenatal testing or predictive testing? That's kind of the highest bar that a particular variant has to meet for you to feel very confident that it is diagnostic. So these are just some of the factors that I've outlined. So given all of this, really how can we communicate these bits of information to patients in a way that makes sense? So just as variant interpretation is standardized, we need to have guidelines and standardization of clinical interpretation of variants. So for this to happen, there has to be a lot of back and forth between the clinician and the labs, and that does happen. But even after that, you probably as clinicians need to have a framework to communicate results to patients. In the Undiagnosed Diseases Network that I'm part of, we have 3 categories of diagnosis that we communicate results in. One is a certain, and that meets all the criteria, and you don't just have no doubt that this is a diagnosis for the patient. In lower levels of confidence, we've got highly likely and tentative diagnosis. A new novel candidate gene that you don't know yet is associated with disease can be a tentative. So this is based on various pieces of information, like what are the characteristics of the variant and the gene, how is it classified, what is the curation of the variant that you yourself have done, what is the current understanding of the disease mechanism and the pathology, and the degree to which the diagnosis might explain your patient's findings. So these are the kinds of things that go into that framework of those diagnostic categories. Why should we use these kinds of categories to communicate results to patient? Well, first of all, it conveys to the patients the degree of confidence you have in the diagnosis. So that really helps them understand better where you're coming from. It also helps with actionability. If you give something as tentative, but make a recommendation of 10 different things that they have to do for their health, they have the ability to question you, well, if this is tentative, do I really need to do all of this? It also places this uncertainty and the fluidity we have with variant classification in a clinical context. So I always say normal and pathological are not a binary, there's something in between. And you can convey that ambiguity by using categories like this. And lastly, it allows for movement for classifications over time. Some things that's tentative today might be a certain diagnosis a year later, or vice versa. Usually the certains don't change, but a likely diagnosis might be bumped down to a tentative and so far. So I think that's the kind of discussion in the community that we need to have. And I'll stop at that.
So I would just say I'm terrified if 75% of clinicians are reinterpreting genetic test reports. No, my experience is they way overcall. So, and that's why I made the point about VUSs. I think they way overcall the significance of a lot of the computational predictors. I mean, ClinGen's actually done a lot of work on this. I highly recommend the preprint that's out there, and I heard the other day it will be soon in press about use of predictors. Some of them are very good. Many of them are way overcalled. But they overcall pathogenic. Many of them overcall pathogenic. They overestimate pathogenicity, but there are some good ones, and there's very little evidence that combining them, which is what the current ACMG rules recommend, is a good idea. But I'm just saying, I think that clinicians are as liable as anyone to want to make a diagnosis, and labs actually, one of the things I've learned from my experience in ClinGen is labs spend a long time looking at these variants. So if 75% of the time they're being reinterpreted, that worries me. The other thing is just a technical issue that has not been well worked out, which is you tell the lab the patient's phenotype, and the physician knows the patient's phenotype. So again, using my cancer experience, child has adrenal cortical carcinoma, highly likely to be associated with TP53. But if I've told the lab that, and they've used that in their interpretation, then I shouldn't use it again. And that's something that's not very well worked out right now for the doctor receiving the report. Did they use my patient's data or only data in the field? So that's more of a technical issue, but I just thought I would comment that I don't have as much faith in people reinterpreting that there should be being done 75% of the time.
And I think you're right, Sharon. It is a large statistic, but that's a reality out there. And I think we need to do a better job at training people in how to interpret this. And that's one of the reasons why I feel like there's got to be a gradation in how results are communicated to families when a clinician is reinterpreting it themselves. You have to be really certain when you call something a certain diagnosis. So not, using loose terms like you probably have this. So this could be the likely diagnosis I think doesn't serve the patient well. We've got to standardize these terms better.
I wanted to share, is this on?
Yeah, I think you just have to get really close.
Okay, so I wanted to share a story as well. And second, what Sharon said. In my experience from a lab perspective, there seems to be an over-reliance on the patient's phenotype on the physician's side. I've had numerous experiences where a really benign variant was questioned because the physician told me, well, my patient has this disease that's associated with this gene. It has to be pathogenic, right? And no, it was not a founder variant in some population. It was just really a benign variant. So I've seen that a lot. So what can we do to educate better, to break that tendency? I don't know.
So do you think just to bring this back to the meta theme of the conference here, do you think that the core challenge in that chasm or that gap between the expert geneticist and the clinician is an appreciation of just the scale of background variation or the scale of variation that exists in all of our genomes?
Yeah, I think it's at least partly what you said. I think people don't have any sense of the scale of variation. And then the other thing that's equally important, and this is somewhere where ACMG was just wrong, is the rarity is actually not a very good argument for pathogenicity at all. Like, it gets moderate strength. ClinGen has recommended down-regulating it to supporting. This is for labs classifying variants. It's actually barely at the supporting level. But we really think if it's rare, and lab reports will always tell you if it's rare, right? That, this must be bad. And, Heidi always points out that I think it's still true, that half of the variation in NOMAD is there once. And not everyone in NOMAD has dozens of different disorders. But so that gestalt, that they have the disease and it's rare, those two things alone just worry clinicians. And I don't know how, because unfortunately being common is a really good argument that you don't have it's a really good argument for being a benign variant, but it doesn't mean that being rare makes it pathogenic. And that seems to be heavily ingrained.
And maybe I'll share a perspective, bringing it back to a more dry topic, the regulation of it. I believe it is underestimated what goes into variant interpretation on the recipient side, because it's been so long practice of medicine when it was simpler, and now there's a lot of work we have to do to even find all the information. And that seems to me a little bit at the core of the matter, and to ensure consistency across the whole spectrum of people who interpret variants, we have to find some way of ensuring that we're all applying appropriate standards. Don't know how yet. I mean, heavy-handed regulation is one approach, but I'm not sure if that's what we all want. So I'm conflicted myself, but what I see, it is still a complete mess, and so something's got to happen.
I would also argue that although if you're saying the literature says 75% of people are reinterpreting, and that's VUSs in specific, but if you look at our own study, there are not many studies that have done this. It's only about 10% of the time that a clinician will question the lab's classification. So most of the time, clinicians do apply, I think, reasonable prudence. And I think that communication between the lab and the clinician is really key to resolving some of those key differences in interpretation.
And I really like the UDN approach. I actually really like the approach of having the physician have to grade essentially the clinical interpretation. I think that's actually a really nice idea. But the UDN is sort of unique, where the clinicians are working very closely with the labs. So the Baylor Genetics Lab, like many labs, is a reference lab. These tests can come from actually other labs. They can come from large hospital systems. They're under more and more pressure, which I'm sure is true of other labs as well, to get reports out quickly. The communication back and forth, and especially, as many of us know, many of the smaller academic labs that used to be in close communication with doctors have essentially closed. They just couldn't compete. And so I do think that communication back and forth is really has really suffered. And they're under the gun to issue a report that goes back into some portal, that goes back to some physician, without there being as much communication as there used to be, or there might be in like the UDN setting or a setting like that.
But I would argue that this clinical classification of diagnoses can happen in regular clinical settings.
Oh yeah, no, I agree with that.
As a community, we need to come up with a framework.
No, I totally agree with that. I just meant this idea that the labs and the docs are going to talk and figure it out just doesn't happen.
So I've been in both settings, small academic, and we were at the time one of the leading forces in this field. And then I transitioned to commercial, and I wholeheartedly agree. I mean, it is critical to have that linkage, but it doesn't exist actually in commercial settings. Now, most providers want to order a cheaper test. And the send-out laboratories in hospitals sometimes make it impossible to send to the highest quality laboratories. I've been there too. Anyway, so I can tell you that in a commercial setting, cost is driven by consumer or provider demand, and it leads to this breakage of communication, because we don't have time. Providers don't have time to send us everything we want. We can't pick up the phone easily and as we used to, so it's a big problem.
But it can happen. In my clinical practice, as well as my colleagues' clinical practice, this is outside of the UDN, we do call the lab. It takes time, you're right.
But what doesn't happen is the lab. So in larger labs, it's more difficult for the laboratory assigning directors to pick up the phone.
I agree with that. But if we communicate, they're pretty good about calling us back.
That's true. The providers that really do want to
Yeah.
communicate, they do. But the inverse is when I used to call and say, really, can you just make sure, can you just tell me if the diagnosis you checked here on the paper requisition is what, what the patient really has? Because I'm seeing XYZ here molecularly. So those communications happen less, initiated by the lab.
So this is a great discussion so far. I just want to take a brief pause and invite anyone from the audience to ask questions at our mics, or anyone from our virtual audience as well in the chat. So maybe I'll ask another question that relates to some of those thoughts around that provider, genetic counselor, genetic testing laboratory communication. So there's been talk, especially I think we saw a lot of discussion on the last panel on polygenic risk scores around how we display information for patients and providers, and whether we're embracing fundamentally a quantitative metric like probability or like an odds ratio for disease, or whether we are sticking to a qualitative categorical label like pathogenicity. So I'm curious, do you see that in the next 5 or maybe 10 years, that penetrance as opposed to pathogenicity will become a core part of the reporting process? Or is this just simply unfeasible for most of the typical scale of Mendelian disease variation that you're dealing with? So maybe Dr. Plon, we could start with you.
Yeah, it's a great question. I think that, first of all, we have to be frank about which diseases do we actually have enough data to give penetrance, right? Because so much of Mendelian disorders have been, based on studying highly affected families. And it's really hard for rare disorders to get good data. I think most labs are already starting to do this with terms like risk allele or moderate, because again, in the cancer space, now there are genes like CHEK2 and things where it really is a moderate cancer risk. We may still recommend breast MRI, but we're not doing more dramatic prophylactic surgery. And so I think there will be gradations of it that we will try to say. The bigger issue, and ClinGen's dealt with this, is we're pretty good at genes where we know a loss of function variant in that gene has a moderate effect on disease. So CHEK2 is a good example of that. Those are still loss of function mutations. They're nonsense mutations, whatever, frame shifts. But the gene itself just doesn't cause that higher risk. The bigger question, which I think is what you're asking, is when different variation causes a wide range of penetrance. And so again, there's some examples. There's the very low penetrant APC variant that if it's reported at all, it's typically marked low effect. I think we want to get there. It is going to be extraordinarily difficult to get enough data, because these are individually rare. These are not genotypes that you can get off of SNP chips, as Carlos was talking about. These are individually rare variants. So to get enough data to say, yeah, we really think this one's low penetrant, that's going to come from massively parallel functional assays. And if we can do those well enough and show that the output from those massively parallel assays correlates with disease risk, then I think we could get there. But it's going to be very difficult to get enough clinical data to say, well, this missense allele is an intermediate cancer risk or an intermediate heart risk compared to this other equally rare missense allele. But if someone's got a really good functional assay that can show is predictive of disease risk, then that's how we may get there.
And that's for the rare ones, right? If you then ask, can we at least do it for the ones like the APC risk allele? I've tried, and we all report them, but when you look at what different labs do, the terminology is all over the map. Another good one is factor V Leiden, where actually research, like what do labs say in ClinVar? And it ranges from benign over risk factor over pathogenic. And so I think we need to come together and build another framework, which is hopefully underway. But I personally think, to your question, it's going to be probably several years before we're doing well with those variants. We're not even doing that great with Mendelian variants, right? So.
Zach Ohane, Harvard. So that was before the previous, in response to the previous question, that was a very interesting discussion about the interaction between clinicians and the labs and understanding the details of it. And I think one subtext I took from that is there's a lot of details to this business that most run-of-the-mill clinicians will not know, and therefore that might be part of why we're overcalling some variants. But there are a group of individuals who are, I would argue, probably a lot more expert than the average clinician, not the average genetic counselor, but the average clinician, and that's genetic counselors. And I'm just wondering, given the growth in the demand for interpretations of genetic data, is there a way forward to empower genetic counselors to order tests, to bill for interpretation of tests, for them to act as the care providers they could be to really scale up to the needs of genomic and precision medicine?
I'll take a stab at that first, if that's okay.
Yes.
Sure. Absolutely, there is a place, and it's already happening in many medical centers, including mine. We have a parallel clinic for genetic counselors only, where they do this kind of thing. They order the tests. The geneticist is involved in a peripheral way, and the genetic counselor is driving the bus. It's very successful. We've had some success with billing as well. And patients are really satisfied with that model. And it's hugely time-saving and allows us to bring in larger volumes of patients who otherwise would just be on that waiting list.
So I recently just looked at the data. Genetic counselors are licensed in 31 states now. I happen to live in a state where they're not, which is Texas. And so that's a huge issue. And I can tell you 10 years ago, it was many fewer than 30. So there's been a huge uptake. For those of you not in the field, there's an unfortunate battle going on in that there is a law being considered by Congress that would include genetic counselors ordering. The various professional societies, including, I believe, the American College of Medical Genetics, is not in favor of that. I work in a pediatric cancer center where PNPs are a key, advanced practice nurses are a key part of the treatment team, and they certainly can order certain tests, and so I definitely think there are models in other fields of medicine where it would make total sense. Ironically, testing laboratories up until recently have been hiring a huge number of genetic counselors, which actually made it a lot harder to hire them and have them in our clinics. I'm biased because I'm in a clinic setting. I think there's a bit of a transition away from that just because some of those labs are having layoffs, and maybe we will get more genetic counselors back in academics, but right now many clinics can't pay as good. So it's interesting that the labs picked up very early that genetic counselors have great skills around variant interpretation. I think if we can train enough genetic counselors and have them licensed, and I firmly believe they should be able to order genetic tests, that it would help. I don't think it will solve the problem that clinician, primary care doctors, oncologists, neurologists, whatever, are still going to be dealing with these test reports for years after it's been ordered. But I think it would be a big help.
Maybe worth mentioning that practice that is becoming more common is that patients take their results, obtain the raw results from the laboratory, and utilize third-party interpretation services that are not necessarily clinically licensed, and then reinterpret these variants. And I've received phone calls, well, you told me my patient doesn't have anything, but I did it myself now, and I found this pathogenic variant. Meanwhile, this is a false positive technical false positives. So that must be very difficult for providers, because I'm hearing that patients come to their offices with that.
We've had a few of those, yes. They do come with that. And one thing that we've discovered is North Carolina also doesn't license genetic counselors yet. But we've been able to work around that by having the geneticist sign off on the testing order. But really the driver of that encounter is a genetic counselor, and it really works well. They do a great job, and it's time efficient. Patients are very satisfied.
The only other thing you can do to help, at least at the front end, that correct orders are being placed that best help the patient's indication is computational support and decision support. But that's not going to happen anytime soon, I think, in any meaningful way. So until then, we need humans with expertise.
Why do you say that that's unlikely to happen anytime soon?
I just don't see it.
Okay.
You know, I mean, and knowing how long it takes to implement even simple things of computational nature in laboratories, I mean, it's conceptually simple to do this, but in practice, it just is hampered by so many resource issues. It's sad.
I mean, there are beginning to be efforts in electronic health records to identify people at risk for Lynch syndrome, you know, sort of the tier 1 disorder. So I think you will see more of that. I did want to point out that the woman over here has been standing at the mic for a long time.
No, thank you all. I'm Cecilia Estevez. I'm with the UDN Coordinating Center here at HMS, and thank you for this great panel. I would love to hear from the three of you as experts in this area. Just going back to the issue of a disconnect between the clinician and the laboratory, in your perfect world, what would that feedback look like with the clinician getting the results from the lab and then actually letting the lab know how they're using those results? So what would that perfect workflow look like for you all?
Well, so for example, the University of Pennsylvania, Kate Nathanson, and many others have actually worked quite closely in the hereditary cancer space with the Epic electronic health record provider and with several large testing laboratories, where they do actually now have, I think, a pretty visionary system where they can order tests. The results come back as structured variants. I think they even have some decision support that it's now triggering. I mean, this was funded originally partly off of a grant, but they're now using it clinically. And I think if you have that kind of system set up, it'd be pretty easy within the medical record to, you know, have messaging back to the laboratory. I mean, I think people more and more, we try to have all of our communication within the medical record. And what's really sad is when we have to go out to somebody else's website and fill out some other form to get the test ordered, or, you know, even use paper forms. So I think if we actually could communicate with the labs through the medical record, you might be able to have physicians be willing to do that, to communicate back. But
can I comment on this?
Yeah.
On the topic of EMR integration, this is absolutely the way to go. Being in a lab that has done a fair amount of that, we integrate with a variety of providers and hospitals' medical records. It is hard because there is no complete standardization yet of those EMRs. So every hospital, every provider has their, own way of doing their own schedule of updating EMRs, and you have to work with all these IT teams across. So, as always, standardization would be helpful to make this a little more seamless.
Great, thank you.
Hi there. My name is Ralph Istanbulia. I'm a medical student here at HMS. So this question is more about the basic science of variant pathogenicity prediction. So my research is sort of about bringing in interactome information, so information about interactions between genes and proteins and all these things in the cell to variant pathogenicity prediction, and the integration is computational. So this is kind of about bringing in the information about epistasis and all these kinds of things to pathogenicity prediction. And I was wondering, considering your experience with as molecular and clinical geneticists, what do you think of epistasis and its role in variant pathogenicity?
Well, first I would say I was an HMS medical student many decades ago, so nice to meet you. We actually talked about this at lunch. Christopher Casa, I don't know if he's still here, a junior faculty member here, was doing some other kinds of predictions, and we were talking about, how can you use them. The easiest thing is if you can actually model known mutations and show that it's actually predictive. And that's, it's not much different in some ways. It's an interesting issue. Is that a functional assay or is that a predictive assay, right? Depending on how much of a lab experiment you're doing. But certainly if you can identify a pocket that you know is important for a key interactor and then be able to map, patient-derived missense variants from there and see if they are predictive, especially for rare disorders, people are desperate for data to help them. And for common disorders, there may be enough data that you could really show that it is predictive. But one of the most important things is to take enough variants to really show that your prediction of the disruption of the interactome is actually predictive of disease. And that's where these things often fall apart. People just pick one or two missense variants and say, see, it works. And there you probably don't, have the power to say that it's really clinically relevant. But certainly there are lots of examples of diseases where we know two actors have to interact and where people have done lab experiments to show, well, that missense variant disrupts that interaction. So doing it on a large scale, I think, is a great idea. It's just getting enough data to show it's predictive.
Thank you. I was going to say it's very reassuring to hear that you've seen this actually in real life. Sometimes like interactions between variants, and so that's good. Thank you.
I had a random question. I hope you'll find it interesting. Let's say that I'm an internist and a patient comes in, a healthy patient. They've gotten their genome done by some particular service, and this is the first time I've ever seen a genome result before. What would be the first thing you could recommend for me as the internist? Thanks.
You want to start?
Sure. I guess my question to you would be was the report supposed to be reporting only things that were diagnostic? Was the report supposed to be predicting risk alleles? I would determine what kind of genome was done. It's one thing if the genome was really focused on reporting rare deleterious variants that are actually linked to rare diseases, where I think the level of interpretation is different. They would report high confidence variants usually, and that kind of patient you could direct very easily if you weren't very comfortable interpreting to a geneticist, for example, that could interpret those results and give them the right advice if they had been diagnosed. Chances of that are low. It can happen. I think where you're going to have more difficulty is when they maybe give you a polygenic risk score of some sort. Those are really hard. In the best of worlds, I don't deal with polygenic risk scores a lot in my practice. I'm somewhat aware of them. I do see some common diseases. They account for a small amount of variance ultimately. And to make really good clinical recommendations on those is really, really hard. So my advice to you is I would seek out whoever did that genome test, figure out what their PRS was based on, what their basis is, and what recommendations do they have, and refer back to them to give you guidance so that you can then get your patient into the right setting that they need to be.
Is your patient unaffected?
Yeah, that was.
Yes.
And does the patient have a family history of any genetic disease that you're aware of?
This is a hypothetical
No, I
question,
just,
but
let's just continue.
there's
So-
one cancer in the family, let's say.
Okay,
Who knows?
so one thing that I would want the internist to be aware of is that it isn't, so laboratories currently often usually issue pathogenic and likely pathogenic variants on these reports, and medicine in the diagnostic era has concluded that they're largely equivalent in terms of actionability. So that's what we're seeing. Oh, likely path means that, I'm going to tell the patient that there's a high likelihood that disease will develop. It's not quite that easy when you do it in a predictive way in an unselected population. Without becoming too complicated, I would send you to a couple of papers that talk about this and say, beware, what gene the variant is in, and educate yourself, if you can, on the likelihood that the positive predictive value of such a likely pathogenic variant can be quite low, actually, depending on the disease. So, I mean, this is one of the things that I see happening now as we're beginning to do more predictive testing, that is an incompletely understood topic in not only physician communities, also laboratory communities.
I think the other thing is really what was the motivation of the patient in getting the genome, right? I mean, typically there's something that they're worried about. And then the question is the genome really the best way to sort that out, right? Versus, you know, like they're actually, you know, the American College of Cardiology does have pretty good risk predictors for heart disease, of which the PRS score helps, but it's not like, you know, black or white. What hasn't gotten brought up today, really, I think hardly at all is, and it's not necessarily totally relevant to your hypothetical question, is now the combination of Mendelian and PRS scores. So this is already occurring now in cancer. I mean, cancer in many ways has led a lot of this testing just because of people's interest and the prevalence of the disease. And so now you have test reports in laboratories trying to combine them. For a while, they would do the like BRCA1 and 2, and if you were positive, then they would ignore the PRS, and if you were negative, they would do the PRS. That was an early model, and they weren't PRS then, they were like 10 SNPs. Now they're actually, some labs are now generating sort of a combined report. And so, you know, obviously in the long run, being able to combine Mendelian risk and population risk is obviously a good thing. But the knowledge of how effective those are and people's understanding of them is, you know, still in its infancy, but they're already being rolled out to patients. And so you might get even a test report like that, and then it's, I think, extremely hard to say, like, you know, does this mean anything? You know, so, but I just wanted to add that that's now starting to happen as well.
I really like this hypothetical scenario, I have to say. I mean, another thing that you, as you're walking away, should consider in this hypothetical scenario is, well, I mean, are you actually well connected enough in the medical community to refer your patient to a specialist?
But for what? They're healthy, right?
Well, for further risk assessment.
Yeah.
Hi, I'm Tom Morris. I'm a software engineer and bioinformatician. Been talking about reports as kind of a point-in-time type of thing here. But while the genome is fixed, the analysis of it isn't. We heard Heidi talk about all the curation work that's going on and everything. So how do you see these reporting mechanisms incorporating the temporal aspect of, you know, when the analysis database or the variant database changes 3 years from now or whatever?
Yeah, I mean, it's too bad Heidi had to leave. She's written on some of this, and she and I wrote an editorial, I guess now a couple of years ago. In the US healthcare system, it's a debacle, right? I recently got an updated test report of a patient I saw 15 years ago when I was still doing adult cancer genetics. I can't even log into that medical system anymore. You know what I mean? Like, but the only name they had was mine, and I haven't moved. So they sent it to me, you know, and so it's extremely difficult in the US system if you're not talking within a couple of years, you know, a year or two. It's one thing if you ordered a test as a physician and 6 months later they got new data and they reclassified it. You're likely still seeing the patient, you can still reach them. But the sort of duty to reclassify, which there have been many different professional societies that have come down on both sides of that argument, I think really has to take into account how chaotic American medicine is. In other countries that have national healthcare systems, they're actually, I think, doing it in much smarter ways, and they are able to put a reclassified report into the national health system data, so whatever physician's now seeing the patient can let them know. But it's extremely difficult in the United States, and labs have policies just totally all over the place. If it's a
that's true.
loss of function variant and that gene is now known to be a disease gene, they'll reclassify it. But if it's a missense variant, they won't. I mean, they're just,
And it affects the PRS as well, because if you rebuild the model, then all the scores change.
oh, yeah.
So from a laboratory perspective, here's a challenge with performing the reclassification, which speaks to your expertise in informatics, right? We don't have, I mean, so most labs have a policy along the lines of, well, every 6 months, if we see the variant again in some other patient, we'll reassess. There's no ability, I can assure you, and despite what others might say, that there's a wholesale reclassification of a laboratory's entire variant database every so often. It's infeasible. So what we need, what I've always wanted, and it's feasible, but it takes a lot of effort, is to have some of these tools that exist now that mine the literature for you in the background, right? And are aware of what types of evidence you need to make an impactful classification change. And it's possible, but it needs to be integrated in the laboratory's knowledge base, variant database, to then proactively send me as a lab director an email saying, hey, there's a paper that just came out. You need to read it. You classified the variant yesterday, so you might think you're safe, but you should actually look at this because it has potential to change. That's the only way we can solve that problem informatically. You know, so.
And I would just say one quick thing. ClinGen has a recuration requirement for our expert panels, and we're already running into how, and we're expert panels. We're not dealing with, we're not dealing with millions of variants or tens of thousands. It's like so much, it's already becoming quite noticeable how much additional work it is for the expert panels, that, we keep trying to update the policy to make sure it's a good policy, but that it's also practical, but that it is quite a substantial burden.
Yeah, I lead one of these panels too. It's a necessary and important policy. It is almost infeasible to implement at scale.
Yeah,
And I
it's a lot
think
of work.
clinical labs sometimes differ. We have 2 labs we deal with. One will occasionally reach out to us, say we had an update with this gene and your patient's variant now has been upgraded. The other lab we never hear from unless we proactively reach out to them and say, look, you classified this 3 years ago, can you do a reclassification now? So I think the onus really is on the clinicians to get those updates.
Unfortunately, right? And so those that don't know, I mean, that's where I think with the regulation needs to come in stronger, because if you wait until it happens naturally, patients will get harmed.
And as Sharon said, so occasionally you get this report on a patient you saw 5 years ago and there's no way of contacting them. They've moved, every known address is no longer valid, and then it's really difficult to
Thank you.
chase down people.
So we're right up at about time. Do the panelists want to give us some final remarks, final thoughts? We started on an optimistic note today. Maybe we can end on a upbeat note.
A final remark. Well, I mean, as I was preparing for this, I was thinking back to when I was young and when the discipline was young. And I was just going to share how much good comes out of discrepancies and divergent opinions. That's really, it's important that we foster that. How it started, to share an episode, I used to work for Heidi Rehm at the laboratory at the time, and in the early days, we would sign out reports, and for a variant that travels in a family, it happens maybe that she signed out the proband, and a year later I signed out a family member and saw her interpretation, and it was not standard. It was pre-ACMG variant classification rules, and the two of us had so many debates over this variant, and she would come to me and say, like, well, I actually disagree with what you said here, and we started putting together Excel tables with what turned out to be the ACMG classification framework in a very, very rudimentary stage. So most people wouldn't know that, but and she eventually led this effort, which I maybe have played a teeny part in. And so anyway, all that to say, controversy is necessary, and we need to just drag it all out in the open to make sure our rules get better and better and bring in computation. computational work.
Great.
Yeah, so I'll try to end on an optimistic note as well. You know, when I mentioned I was a medical student here actually long before this building was ever built, but when I was first a medical geneticist, we did not order sequencing on any gene. BRCA1 and 2 were really the first. Actually, APC, those of you who are young don't realize the way we used to do APC testing is they would make cDNA, they would make RNA, they would in vitro transcribe it into little peptides, and they would look to see if the peptides were of different lengths. Protein truncation assay. And if you had a patient who had a little peptide that was of the wrong length, they had a pathogenic variant in APC. I personally have since seen the children of those patients, and we had to resend the proband's test to say, could you now tell us what the mutation is? But it was too expensive to sequence then. Even if you knew the segment, they didn't sequence. So when I was working with the Baylor Laboratory and the Genome Center to develop the exome, one day I realized, oh, I can read these exome reports on my cell phone. So I literally went in my own career from not being able to order sequencing of a small peptide where I knew a mutation occurred from looking at the results of a patient's exome on my cell phone, which of course, who knew what a cell phone was? So I think we should be optimistic of where technology is going to drive us, but I think we really have to be realistic about the load on physicians and come up with ways to communicate that so that the information's useful and not overwhelming or misinterpreted.
I agree with you. I share Sharon's optimism. I started genetics practice back in 1995. I could diagnose a handful of patients in my clinic. You know, chromosomes, then came FISH, then came subtelomeres, and sequencing has made a huge paradigm difference to my practice and my research. So I think we know more. We're also better about knowing what we don't know and the limitations of what we do, and translating that into practice, as well as making sure we give our patients information that is useful to them and is just not confounding to them. And being very clear about what we know and don't know about their results. I think that's where we need some work.
All right, thank you. So please join me in thanking our third panel. All right. Okay, all right, we did it. We made it to the end of the day. I promise I will be brief with the closing remarks. First of all, I want to thank my co-organizer, Zach Kohani, our two keynote speakers, and all of our moderators and panelists for discussions that I think were both illuminating and very engaging. I also want to acknowledge the support of our sponsors, Takeda and Merck. And I'd like to personally thank the DBMI staff, the DBMI team, and particularly Samantha Lemos, without whom I think this event definitely would not exist. And I want to thank all of you, both in person and our virtual audience, for tuning in and for engaging with our speakers. So I think just reflecting on the themes of the day, I think at first blush, it seems like it's an easy task, right, to define what is a normal genetic variant result, a genetic test result, or if you're thinking about a patient's or your own blood test results, what a normal result might be when you visit the physician. So we measure something in a reasonable number of people, and then we flag someone if they fall outside of that typical range. So I think what's come up today over and over again is that there's a lot of questions that emerge as we are trying to actually do that seemingly easy exercise. Who exactly is this normal population? How many individuals are enough? From what backgrounds and from what population groups? Which variants are benign? Which variants are pathogenic? Will transformative new technologies like CRISPR that we heard about in the morning reach a small or large overall fraction of the burden of disease? And what is causation? What is correlation in our models and in these reports and in these labels that are changing lives and governing care every day? I think many of the speakers that we heard today have had real clinical impact because they haven't shied away from these questions. They haven't left them for someone else to answer, but they've tried to answer them themselves in the context of life-changing and very important genetic testing and genetic technology. I think deepening our understanding of normality is central not only to genetic testing, as we heard about today, but to many areas, basically all areas of clinical practice. We're always answering this question: what is normal, what is abnormal? And I hope that the perspectives that you heard today will help you enrich your understanding of normality and for us all to better achieve precision medicine. So thank you again, and I hope you'll join us next year.